Hello everyone, in this presentation I am going to present on the topic preparation and in vitro evaluation of PLG nanoparticles of esomeprazole for oral delivery. This practical work has helped me gather knowledge about the different method of preparation of nanoparticles and its various method of evaluation. Introduction. Septic ulcer is one of the most prevalent diseases worldwide, affecting almost 10% of the human population. Septic ulcer are of two types, gastric ulcer and duodenal ulcer. The most common risk factor of septic ulcer are stress, bacteria like H. pylori, consumption of alcohol, and smoke. To treat such disease condition, proton pump inhibitor are the most promising molecule, and esomeprazole is one of the proton pump inhibitor which act by inhibiting the hydrogen potassium ATPase enzyme, thereby stops the secretion of hydrochloric acid. Hence, this proton pump inhibitor can be formulated as a novel drug delivery system in the form of nanoparticle by emulsification solvent evaporation method to obtain esomeprazole loaded PLGA nanoparticles. Objectives. The main objective of this work is to prepare and characterize nanoparticle of esomeprazole and to improve the esomeprazole loading in the PLGA nanoparticle, which was the most challenging task, and to perform in vitro evaluation aiming to develop an extended release formulation of esomeprazole which has a shorter half-life of 1 to 1.5 hours. Method of preparation of nanoparticle. Nanoparticle was prepared by emulsification solvent evaporation method. At first, the polymer was dissolved in a binary solvent and sonicated to dissolve the polymer. Then the drug was added to the polymer solution, which was also sonicated to dissolve the drug. And the drug polymer solution was added dropwise in an emulsified solution under homogenization to obtain the emulsion. And the obtained emulsion was transferred to a round bottom flask which was set in a rotary vacuum evaporator to evaporate the organic solvent. After the evaporation of the organic solvent, the emulsion was centrifuged to collect the nanoparticle, and the nanoparticle were collected in a petri dish and was freezed. After freezing, the nanoparticle were lyophilized to collect dry nanoparticles. Characterization of nanoparticles. Nanoparticles were characterized by particle size distribution and zeta potential in Antor particle size analyzer. Then surface morphology was studied by FSM and ANSEN. Functional group analysis was done in FTIR. Encapsulation efficiency and loading capacity was determined in UV visible spectrophotometer at maximum absorbance of 301 nanometer, which was of the pure drug obtained by determining absorbance maxima of the pure drug in phosphate buffer solution. Then in vitro drug release study was done using dialysis membrane diffusion method. Result and discussion. The particle size distribution and zeta potential was done in DLS. Here we can see the formulation table of different formulations with their average particle size zeta potential. Column 1 represents the 7 different formulations. Formulation F1 is a blank nanoparticles. Then formulation, then column 2 represents the polymer drug ratio. Then in the table, column 3 represents the organic solvent used to dissolve polymer and drug. Then column 4 shows the aqueous non-solvent used for the formation of emulsion and the stabilizers used in different formulation. Then the last two columns shows the average particle size and zeta potential of the formulation. Here we can see the formulation F7 shows better average particle size and zeta potential as compared to the other formulation. Zeta potential is the measure of surface charge of a particle. If zeta potential is less than -30 or greater than +30, then the formulation is considered to be stable. Here in the figure, we can see the particle size and zeta potential of formulation F7. Then we come to the surface morphology. The study of shape and morphology of the freshly prepared nanoparticle were carried out by FSM and SEM. Here in the figure, we can see the SEM and FSM image of formulation F7. Then the functional group analysis. In the present study, drug excipient interaction study was carried out by FTIR spectroscope. FTIR spectrum of pure esomeprazole was characterized by the absorption band of different functional groups, and the FTIR spectrum of different formulations were also recorded and compared, and it was found that the absorbance band were identical and there was no change in the functional group absorption of any of the molecule present in the formulation. And hence we can say that the formulation prepared were stable and no chemical interaction took place between the drug and the chemicals. Here we can see in the image uh, FTR spectrum of formulation F7. Then the encapsulation efficiency and loading capacity determination. Encapsulation efficiency and loading capacity of a nanoparticle greatly depend upon its content, like polymer concentration, method of preparation, and drug use. Here in the table, we can see the different formulations with their binary organic solvent, percentage drug loading, and encapsulation efficiency. The formulation F7 shows better drug loading and encapsulation efficiency as compared to the other formulations. Then finally, we have done in vitro drug release study. For in vitro drug release study, 2 ml of the nanoparticle was reconstituted in 2 ml of phosphate of solution and taken in a dialysis membrane. Then this dialysis membrane was tied on its both the ends with a thread. Prior to the experiment, this dialysis membrane was soaked in a phosphate of solution for 12 hours and rinsed with distilled water to remove the preservative. Then this assembly was immersed in a 100 ml beaker containing 50 ml of phosphate buffer solution, which was used as a release media. And this setup was set on a magnetic stirrer at 300 rpm. And at a predetermined time interval, the sample were collected and replaced by fresh phosphate buffer solution to maintain the sink condition. And the sample was analyzed in UV visible spectrophotometer and the concentration was calculated using calibration curve. An image of in vitro drug release study can be seen here in the figure 5. Then we come to the table where we can see the different kinetic model and equation for formulation F7. Formulation F7 was considered for in vitro drug release study because of its better drug loading and encapsulation efficiency. Here in the right hand side we can see a graph which shows the log cumulative percentage of drug release versus log time as Cosmer-Pipas model. This model helps us to determine the release profile of a polymeric formulation. Then we come to the conclusion and future prospect part. Esomeprazole loaded PLG nanoparticle was developed by emulsification solvent evaporation technique. Nanoparticles were characterized by different methods and these data were used to evaluate the different formulations. Here we can see the percentage of drug loading and encapsulation efficiency for formulation F7 was much better as compared to the other formulations. However, the percentage of drug release was 80% over 6-hour release period. The in vitro drug release suggests that esomeprazole would release in a sustained manner from the nanoparticle formulation over a prolonged period of time. And in future, development of esomeprazole loaded PLGA nanoparticle may be entry-coated for better formulation development suitable for oral delivery of esomeprazole to the gastrointestinal tract for the treatment of peptic ulcer. These are the references with the help of which I have been able to complete my project and this presentation. I would like to thank my mentor and my friends for guiding me throughout my project and help me for the completion of my project. And I would like to thank DRPI for giving us this platform for presenting our project work. And I would like to thank all of my viewers for viewing my presentation. Thank you very much.